Hi everyone. Before we begin, we'd like to thank the Maestro family and the Brain Tumor Foundation for this opportunity. Today we'll be discussing intranasal administration of telomerase inhibitor GRN163 as adjuvant therapy to post-surgical resection in diffuse astrocytomas. So a diffuse astrocytoma, or DA, is a grade 2 glioma and is characterized by the fact that it's slow growing and has a median survival rate of 5 to 7 years. The current standard of treatment is maximum surgical resection followed by chemotherapy and radiation. This is proven to be ineffective because total resection is not normally possible due to the infiltrative nature of the tumor. And because of that, there's a higher rate of recurrence and/or malignant transformation. Furthermore, it's ineffective because of the low mitotic rate of the tumor. So because patients undergo chemotherapy, prolonged exposure to chemotherapy and radiation, it has a negative consequences on their quality of life.
Okay, so next we'll discuss the role of telomerase in cancer. So the ends of our chromosomes are protected by long stretches of repetitive DNA called telomeres, and telomerase is the enzyme which is responsible for maintaining their length. In normal cells, telomerase is rarely expressed, and thus telomere shortening causes the cell to either undergo apoptosis or to permanently stop dividing. In contrast, in cancer cells, the overexpression of telomerase confers cellular immortality, and thus if we were to inhibit telomerase, we would be inhibiting proliferation as well as inducing cellular death.
The main complexity in treating brain tumors is the blood-brain barrier, which controls the movement of substances into the brain, thus disallowing the majority of cancer treatments from entering. Thus, we have decided to circumvent the BBB intranasally because it does not require to bypass the BBB to reach an astrocytoma. So, other advantages include the avoidance of systemic circulation, which then reduces the risk of side effects. It is also painless and non-invasive, and the rapid action of treatment, as well as the self-administration by patients, would reduce the possibility of long-term administration in hospitals. Furthermore, because GRN163 measures 13 amino acids, it would follow the olfactory pathway to the brain. Thus, the passage begins at the olfactory receptor neurons, which then projects to the cribriform plate, into the cranial cavity, and then onto the olfactory bulbs. The olfactory bulbs then project to the brain parenchyma, the spinal cord, and the cerebrospinal fluid. Furthermore, GRN163 can reach any portion of the brain, thus any tumor in the brain, because it can reach as far as the occipital lobe and the cerebellum, which means that it has access to all of the brain. So why did we pick GRN163? Well, first of all, GRN163 is an oligonucleotide, which means that it is complementary to the template region of the human telomerase RNA subunit HTR. And basically, contrary to chemotherapeutic agents, telomerase inhibitors target tumor tissue, leaving healthy tissue unscathed. And because the delivery of GRN163 is precise, it targets telomerase, which is expressed in 85% of human cancer cells. Thus, treatment with GRN163 has been shown to induce telomere shortening, growth arrest, and cell death in a prostate cancer xenograft model. Furthermore, the lipid conjugate of GRN163, which is known as GRN163L, is currently found in clinical trials in phase 1 and 2.
The administration of GRN163 would be an effective adjuvant treatment following surgical resection. This is due to its ability in inhibiting tumor growth, thus eventually increasing survival rate of patients. Our specific aims are to design an effective and non-invasive treatment of bypassing the BBB, improving the progression-free survival rate of patients, and providing Connie with a viable clinical trial.
So next we have two preclinical studies involving GRN163, both of which were conducted in rodents bearing intracerebral human tumor xenografts. So the first study demonstrated that tumors treated with GRN163 grew significantly slower than the control group, while the second study demonstrated that the intranasal administration of GRN163 over the course of 12 days significantly prolonged the median survival rate of the treatment group.
So we propose a phase 1 clinical trial in order to evaluate the safety of the method, find a recommended dose according to the individual's metabolic rate and brain tumor size, as well as to look for early signs of effectiveness.
We require diagnosis of primary DA, partial surgical resection of DA. Participants with prior DA treatments, such as rhinitis or chronic respiratory diseases, will be excluded.
So thus, we're proposing a single-arm, open-label study in which GRN163, formulated in a saline solution, will be administered intranasally. To determine the maximum tolerated dose, we will be using the traditional 3+3 rule-based design. The starting dose will be determined from animal models, as well as adjusted for metabolic rate and tumor size. It will be administered into both nostrils to increase absorption and adapted for the nasal mucosa consistency of all patients. A follow-up post-treatment will be conducted. So the primary outcomes of the study will be to determine the maximum tolerated dose of GRN163, as well as to determine the safety of the treatment by the evaluation of adverse events, hematological assessments, urinalysis, and physical examination.
For the secondary outcomes, we'll be testing the efficiency of GRN163 in terms of tumor size and telomerase activity. So for the tumor size, we'll be using periodic MRI scans, and we will also be calculating the differences in tumor volumes using baseline and post-treatment scans, as well as different dosage groups. For the telomerase activities, plasma samples will be collected to test pharmacokinetic effects, and PBMCs will be collected to test pharmacodynamics by TRAP assay.
Reduction of the tumor size and inhibition of the rate of growth will allow for an increase in progression-free survival rate. Decrease in telomerase activity in the peripheral blood mononuclear cells will also be observed. And lastly, there will be an established safe dosage which will be adaptable for each patient.
So we just have like 2 more seconds to go, so sorry. So this particular study is innovative and supportive of the goals of brain tumor research because it provides safe methods for patients and for therapy. So this also provides new alternatives, and it would provide like new treatments in general to patients, as it became for Connie, our case study patient. And finally, we'd like to have some acknowledgements. So first, we would like to dedicate this treatment and presentation to my father, who I lost to brain cancer in January 2017. And then we would also like to thank the Brain Tumor Foundation and the Maestro family for having us here today, as well as the judges, and all of you here, all of the audience, both here and at home watching, for your attention throughout our presentation. Finally, we would like to thank Dawson College, Concordia University, and McGill University for their guidance and for their help throughout this process. So thank you so much for having us here today.
Great work, ladies.
Thank you.
Now for the hard part. So
Lovely.
you mentioned something that I need a better explanation of. So you mentioned testing telomerase activity through peripheral blood.
Yes.
However, your study design is specifically designed not to end up in peripheral blood, based off of the way you explained it at the beginning.
Right.
So the expectation, based off of clinical trial design, is that you would see no difference in the peripheral blood telomerase activity. So can you walk me through how you would test the telomerase activity if by study design you shouldn't see any? Is that just a safety factor? Because that's not the way you presented it.
Right. So it is a safety factor, but also because peripheral blood is more likely to be affected by disease in general, which is why it's used in immunology for the most part. We would be testing baseline blood samples for their telomerase activity, and despite GRN163 and intranasal administration reducing the risk of systemic circulation, we would nonetheless see it. I mean, it's bound to happen, but because telomerase only affects telomerase inhibitors, only affect telomerase, we should only see an impact on the telomerase activity, which means that through this peripheral blood, we could see a change in telomerase activity, despite our treatment trying to avoid peripheral blood, because it's bound to happen. We would like to avoid systemic circulation.
So I'm with you.
It's more for a safety purpose, yeah.
Yeah. So 2 parts, and then I'll let you move on.
Perfect.
One, that's not the way you would never get that clinical trial passed. You cannot have a test that is designed to test for something because you think it's gonna go wrong, right? Because by design, your study is designed to not have that happen.
Right.
And the other part is when you're presenting something like that, you want to couch it in if it's a safety concern, you couch it as a safety concern.
Okay.
Yeah.
Thank you.
Thank you. I enjoyed your talk very much.
Thank you very much.
It's an interesting, novel idea.
Thank you.
I think. The intranasal route or transnasal route has many advantages, I think, over other routes of delivery of medication to the brain. But what advantage is there of this over other methods of disrupting the blood-brain barrier? Because there are many. So why this particular route?
That's an excellent question. And the reason for that, I believe, is that I think we can control for a few more variables. And also, it is really an excellent question. And I think that the reason why I will finish answering your question. I think that because we are avoiding disrupting the BBB completely, right? We're passing through a different route completely. We're passing through the olfactory bulbs, and so we're avoiding disrupting the BBB. This means that there's less risks also of perhaps another microbial virus of some sort to enter as we're disrupting the BBB, because we're not even touching the BBB. We're completely avoiding it. So I think that this method provides advantages over other disruptive methods because, we don't even need to go through it.
Thank you to Team 3.
Thank you so much.